Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the principle of crossing over. What we'll do in this video is leverage that principle to see how we can calculate recombination distances between genes. And later you'll see how we'll actually use that to construct a map of the genome, or a map of multiple genes. Let's first recap some of the points from the last video, just to remind you. Again, we can use linkage to develop a map of chromosomes. This is our ultimate goal. Now, importantly, as we said before, chromosomes are typically linear. Neighboring gene variants tend to remain associated even when you have crossing over. So if you have two genes that are very close together, you'll tend to inherit alleles of those genes together. Whereas genes that are further apart, you may be more likely to get recombinant forms. If they are very close together, you typically only get the parental types. If they're further apart, you may get some parental, some recombinant. So what you tend to see from this is the full range of possible associations. You may have no recombination or no crossing over, particularly in this case, between two genes. This would mean they're very, very close together. There's nothing breaking them apart. Or you can have no association, basically 50% recombination. This is what you see when things are very far apart on a single chromosome, or you tend to have them on different chromosomes, one of those two things. At that point, they are independent of each other if you get 50% recombinants, 50% parental. Now, let's start by looking at how we identify what's going on along these chromosomes. So I was depicting these chromosomes before and referring to points A, B, and C along them. But how do we know what variant is at A, what variant is at B, or what variant is at C? Well, the answer is we use genetic markers. The genetic markers are basically reference points in the genome that have two alleles or more. They have two forms or more. Now, you don't necessarily know where this is. Now, this sounds a little bit funny because you can actually generate a map without knowing where at all you are. You could think of it in the context of saying you know where something is relative to something else, but you may not know exactly where that is. So, for example, I could tell you there are three genes, ADH, ACT, and ALDH. And you can say that I know that ALDH is in the middle of the other two, but you don't know what chromosome any of them are on. That's okay. That's the start of how you put together a genetic map. Now, genetic markers can be molecular, right? Being molecular, meaning that you assay them using some sort of molecular biology technique, such as, for example, doing PCR, or the polymerase chain reaction, and assaying it on a gel. This depicts right here, the bottom left picture, different alleles. So this could be a big A individual. So that's two copies of the big A allele. This is a little a individual. That's two copies of little a. And here is your heterozygote, has one of each. Right? So this is a very easy assay there. In this particular case, what's happening is they run differently on the gel, that the smaller fragments migrate faster than the larger fragments. And there's a difference in size where the little a has this extra set of bases in it that's not present in the big A. And that makes the little a run slower down the gel and tend to be higher up. That's just one example. Don't worry about the details of this, but this just shows you what we mean, or how people will assay a genetic marker. Now, genetic markers can be molecular, but they don't have to be. They can also be phenotypic. They can also affect the appearance of an organism. So we talked already about these cases of having red versus white eyes, these Drosophila mutations. Now, you can say, for example, this red-eyed individual may be big A, or big A, little a, because we know that the red-eyed one is dominant. This white-eyed female may be little a, little a. Now, there's obviously some underlying nucleotide difference, some underlying DNA sequence difference at an eye color gene that's making it so the little a individuals have white eyes. But we can nonetheless use these as a genetic marker. And in fact, this is something that was used extensively over the course of the last century, and still at some level today is used for making genetic maps, these sorts of phenotypes. This is also how we map genetic diseases, and we'll come back to that a little bit later on. Now, what we do is we use either controlled crosses or pedigrees. They're basically the same thing. Controlled crosses being when you take the red-eyed fly and mate to the white-eyed fly, or pedigrees is you're looking at things that have already happened and just inferring what's going on. So you can say that we know your great-grandfather had brown eyes, but we know your great-grandmother had blue eyes. And look at what's happened. We infer the genotypes to make a map. Right? We infer the genotypes to make a map. So let's use the case of what's referred to as a test cross. Okay? So what we want to do is we want to identify parentals and recombinants. So we'll start with something which is homozygous, which has only one type. Big A, big B, little b, crossed to little a, little b, little b. Okay, so let's say the red is the female and the blue is the male. We make this offspring, right? So this is a heterozygote, big A, big B, little b. We know this is the phase. We know that you got the top one there from mom. We know got the bottom one there from dad. What we do is we cross this to the homozygote again. So cross it to a, little b, little b. Now, if A and B as genes are totally linked, what fractions should we get from all these different things now? If there's no crossing over between A and B, what should we see? Look at that for just a second. If there's no crossing over between A and B, we'll only get the parental types. So in this case, the parental types will be big A, big B, this set, right? And little a, little b, which is this set. And then this individual always gives a, little b. These would not be found at all. If they were totally linked, you wouldn't get any of those. Okay, now let's do a different test. Let's say they're completely unlinked. Let's say they were even on different chromosomes. What would happen then? If they're on different chromosomes, you get all four types. And in fact, you would get 1/4 of each of these. So these are the parentals, these are the recombinants. Half of the overall offspring would be recombinants. These are your recombinants. Now, the question that I want you to think about is what happens if, somewhere between these two extremes, not the totally linked and not the totally unlinked, like being on different chromosomes. What happens if they're sort of linked, like they're near each other but not right on top of each other? Well, what ended up happening in that situation is you get mostly these, but you get some of these. Basically, this would be more than 50%, but less than 100%, and this would be something less than 50%, but more than 0%. What this recombination fraction tells you, what the fraction recombinant tells you, is how far apart the A and B genes are. The further they are apart, the more the percent recombination will approach 50%. The closer they are together, the more the percent recombination will approach 0%. So let's look at this using this particular fly cross. Now, here what we'll be doing is we'll be crossing flies that are mutant for one gene to flies that are mutant for another gene. So let's start with over here with these ones on the left. These ones have cinnabar or bright red eyes. So this is different from the standard fly eyes, which are more of a sort of a dull red. And let's say we're crossing males, your boy symbol, to females that are vestigial. Okay? Vestigial has these sort of shriveled up wings rather than the normal full-length, beautiful Drosophila wings. Now these boys here have normal wings. So we can say that they are cinnabar, so they have the CN, but they are VG plus because they have the normal copy, not the mutant copy of this. In contrast, these vestigial winged females, we can say they are CN plus because they have normal dull red eyes. They don't have the mutant bright red eyes. So we're crossing CN VG plus males to CN plus VG female. Okay? So these ones here are mutant for cinnabar. You're mutant for the thing that doesn't have the plus. So we're crossing these mutant red-eyed males to these mutant squished up wing females. Okay? Now, we cross these and the offspring all have normal eyes and normal wings. So what can we infer from this? Well, we can actually infer quite a bit. First is the cinnabar form, the bright red eye form. Is that mutation dominant or recessive? Right? In this case, we know the mutation has to be recessive, right? Because we crossed a fly that was cinnabar to a fly with normal eyes. And in the end, the flies all had normal eyes. So normal must be dominant or cinnabar must be recessive. Similarly, vestigial must also be recessive. Because again, we crossed a vestigial winged fly to a normal winged fly, and the offspring all had normal wings. So normal wings is dominant, or vestigial form is recessive. So what would be the genotype of this fly? Remember we said that this one over here was starting off with CN VG plus. This one was CN plus VG. Well, the genotype for this fly, when we're looking at basically the combination of alleles it has, would be something like this, where it has the CN VG plus from one parent, and it has the CN plus VG from the other parent. This is the combination. Now we're going to assume that these are actually on the same chromosome, which actually they are on the same chromosome, but let's just assume that we're guessing that. Now let's say we cross F1 females. So these are what's referred to as the F1 females. Okay, these offspring up here. Let's say we cross them to males that have the Cinnabar mutation and the vestigial mutation. Well, what would the genotype be for these males? If they have both mutations, we know it's recessive, what would the genotype for them be? Well, since the males were exhibiting both mutations, the male is necessarily homozygous for CN and VG because it has cinnabar eyes and vestigial wings. Okay? So we're looking at, these are the males that we're going to cross them to. These are the F1 females right here. What would be the parental and the recombinant offspring? What would they look like? Well, let's think about this. This is always giving the mutant forms, right? So the dad's always giving the mutant form. So basically from dad, you're always getting CNVG, no matter what. You're always getting that combination. It doesn't matter if there's a recombination or not, which there isn't any recombination in Drosophila males, but let's just leave that alone. If you're getting parental gametes from the female, what would you get? You would get either CNVG plus, or you would get CN plus VG. So what would these flies look like? Well, this top set of flies here would have the vestigial wings, so they would look like this parent. This bottom set of flies would have cinnabar eyes, so they look like this parent. But that would be it. You would not have any normal-looking flies, and you would not have any flies that look like their dad, that are mutant both. Recombinant offspring would get something like this. They would get the CN and VG from mom, which they'd also get a CN and VG from dad, meaning they would be mutant at both genes. Or they would get CN plus and VG plus from mom, meaning they would look totally normal. They would not have any mutant. So let's look at these. Here are all four possibilities. These top two depict what would happen in the context of just getting parental types. The bottom two depict the recombinant form. So what if there was total linkage? What if the CN and VG genes were right next to each other? What if there's total linkage? What would you get? If there's total linkage, you'd only get these two forms, right? If CN and VG were completely unlinked, you would get half these forms, half these forms. So maybe a quarter of each of the lines there. But what if they are linked but not totally so? That's when you get mostly these and a little bit of these. Okay? Now, again, it's this fraction recombinant that reflects the distance between genes. That if genes are very close together, then the fraction recombinant will be low. If the genes are very far apart, the fraction recombinant will be high. Now, Thomas Hunt Morgan, who I mentioned, the Drosophila geneticist, he suggested this percent recombinant can be called map units. And often now in Drosophila, they are referred to as centimorgans in honor of him. So again, this recombination fraction ranges from 0% to 50% recombinant. And this is often just referred to as 0 to 50 centimorgan. Okay? Now this gives you an idea of the distance between genes. And this was known like 100 years before we had the first genome sequence. Not quite, but close to it. And at this point, we had a way of putting together a map and saying, this gene is close to this gene, this other gene is really far away. And the genes in this case were things like these Drosophila mutations, like the cinnabar gene, the vestigial gene, the white eye color gene, things like that. In humans, just for your reference, 1.3% recombination is on average about 1 million bases. That's not exactly the case, but that gives you sort of a ballpark that you can work with. And this is used to determine the linear order. And we'll come back to how that's done in the next video. But let's put in some numbers here. Let's say that you were counting flies. So this top fly here has normal eyes but vestigial wings. The next one has cinnabar eyes but normal wings. The next one is normal at both, and the bottom one is mutant at both. Now in this case, they all have the mutant copy from their dad. This is that test cross. So they're all over CNVG. Because they are diploid, they do have 2 copies of everything. But what I drew it on here was just to depict the phenotype or how they look. Let's say you got these numbers. What does this tell you? Well, these are all the parental types, right? These are the ones that match. Basically, they got this this gamete from their mom or they got this gamete from their mom. So these are parentals. These lower ones here are recombinant because they had to get either this form or that form from mom. So these are the recombinants, these are the parentals. Yeah? Now what fraction overall here are recombinant? Well, the total here, the total number of flies here is 200. The total number of recombinant here is 20. So the recombination fraction is 10%. There's 10% recombination between these two things. So this gives you some idea of distance between these. Now, in the next video, we'll show how you leverage this to generate a map, and particularly using 3 or more genes and identifying the linear order. In the meantime, look online for some sample problems that you can try on your own. Thank you.